Hello again, my name is Nur Irdina binti Nosri with the metric number of 146806 from Bioprocess Division and from School of Industrial Technology. I am going to present you the lab practical video 4 with the title of Subculture of a Cell Line and Counting Cells with a Hemocytometer. Enjoy watching! The first video I am going to cover is the subculture of a cell line. The purpose of subculturing of a cell line is to maintain the optimum conditions of the cell cultures, which are crucial to keep the cells in its log phase of growth. The type of cell line we are using in this method is adherent cells. The cells will keep growing in vitro condition and until it has covered the surface available to grow or the nutrients have decreased in the medium. The chemicals and equipment involved in this technique is complete media, trypsin or EDTA, phosphate buffered cell line or PBS, an inverted microscope, pipette, flask, sterile container, and centrifuge. Firstly, the cultures were assessed by examining the condition of the cell monolayer by inverted microscope. This is actually to ensure that the cells were healthy and are able to undergo subculture. Secondly, the spent medium is removed from the vessel. Thirdly, the monolayer cells were washed with warm phosphate buffered cell line or PBS twice. Next, the warm trypsin or EDTA was pipetted onto the monolayer cell to detach the cells and can be monitored by inverted microscope, and the plate was left to approximately 5 minutes at 37 degrees Celsius. It is important to not expose cells to trypsin or EDTA more than 5 minutes to avoid damage of cell receptor. Moreover, the flasks were gently tapped to detach the remaining cells and a warm complete medium was added by using pipette and were pipetted up and down to break clumps. The cell suspension were then removed from flask and pipetted into a sterile container. Then, the detached cells were incubated for 2 to 10 minutes. After that, the cells were centrifuged at 1000 rpm for 5 minutes. The supernatant was removed from the flask. The pellet at the bottom of flask was resuspended with complete medium by pipetting it up and down. Lastly, a viable cell count was performed and a liquid of cells was reseeded in a flask with the desired density. A complete medium with suitable volume was added into the flask and it was labeled according to its cell line name, passage number, and date. The second video is counting cells with a hemocytometer. Mind you that I will attach the video related to counting the cells with hemocytometer since I don't have the equipment suitable at home. So the purpose of counting cells with a hemocytometer is to determine the quantity of cells due to usage in cell culture testing. Trypan blue stain is utilized as to distinguish the living cells and the dead ones in counting. The chemicals and equipment involved in this technique is 0.4% of trypan blue stain in phosphate buffered cell line, cell suspension, and hemocytometer with cover slips, pipettes, and inverted microscope respectively. Firstly, the glass hemocytometer and cover slip was cleaned with alcohol. Secondly, preparing cell suspension by swirling the flask gently to make sure an evenly distribution between cells in the flask. An amount of 0.5 ml of cell suspension was pipetted by using a sterile pipette and placed in Eppendorf tube. Thirdly, an amount of 100 microliter of cells was pipetted into a new Eppendorf tube and 400 microliter of 0.4% trypan blue with a concentration of 0.08% was added and mixed gently. The rest I will let the reference video to explain how they count the cells by using a hemocytometer. Thank you.